All right, hi everyone. We're here today for a quick video showing you guys the basics of how to make a smear on a microscope slide and how to then stain that smear. Well, how to fix the cells on that slide so that they're killed and fixed in place, and then how to stain them so that you can actually do some microscopy and look at them under a microscope. So we are going to get started. You've gone over the theory of this, so this is how you actually do it in the lab. So Sabrina's here, she's got her Bunsen burner going because the first part of this, you're going to need your aseptic technique still because you're working with live cultures. We're gonna start off showing you guys how to make a smear from a broth culture. So we have here a broth culture of E. coli, it's nice and turbid. So cloudy there, and Sabrina's getting her loop flamed and ready. And then we're going to show you guys how to, you can see the slide down there, and I'll zoom in a little bit so we're closer in. And there's the slide there on the laboratory bench. It's got a tape marking at the end. And Sabrina, actually no, we're gonna put the smear on first. We'll go over what you put on that tape in a minute. But right now she's going to open up her tube, flame the neck of the tube, reach in with her loop, pull out a loop full of bacterial culture. Then she's going to go down to that slide, and what you'll see is she's going to smear out about a nickel-sized area. It doesn't really matter exactly how big, but you don't want to go above a quarter, and I wouldn't necessarily go below a dime in terms of the size of what is really exactly what it sounds like, a smear of bacterial culture all over that size of circle on your slide. You want to put it right in the center of the slide. You don't want it way off hanging at the end, that smear of bacterial culture. And that's all there is to making a smear from liquid culture. We'll show you the next steps just to get it dried faster. But right now, Sabrina's going to show you how to set up one from a solid culture. So if you're picking from a colony on a plate, and she's actually using a wax pencil, which is a very handy way of kind of marking where you're going to be putting your smear. I definitely advise that because sometimes you think, oh, I put it in the center, but you can't quite remember. It's nice to know for sure where that smear is going to be. So there's the slide down there. You can see the circle on it. I'm just going to zoom in a bit more for you guys. And she is just going to get some of that water off. So you want a little drop of water on there so you can mix in your solid culture and get it kind of mixed evenly in a drop of water. What can be a little frustrating sometimes is these water bottles give you a giant squirt of water. But now we have there, and you can see it kind of in that black ring there, there is a nice drop of water there. And this is where we're going to now get some bacteria into that drop of water. So you'll see we didn't use sterile water here because we're actually going to be we're actually going to be killing these bacteria anyway. So it doesn't matter whether that water is sterile at this point. So Sabrina's taking a culture or a little bit of a colony from a plate of E. coli there, and she's smearing that around. You can see it turns it immediately a bit cloudy because of the bacteria that are coming from that colony. You do not, if the colony is very, very small, you might lift the entire colony off the plate. If the colony is quite big, as those colonies were on that plate, you really just need a touch. A little tiny piece of that colony is all you need. Just flame the loop to sterilize it. And she's now moving over, and you can see kind of a black rectangular box down there. This is called a slide warmer, and she's placing the slides on the surface of that slide warmer, and it's exactly what it sounds. It is heated to about 50 degrees. It is warming those up, and it's helping speed up the evaporation. Because the next step in our smear technique is to get rid of all the water. So the liquid from the culture, and also the water from the water droplet you put on there from the squirt bottle needs to evaporate so that you're left behind with kind of a thin film of bacteria on there. So I'm going to stop this video now because we're going to wait for those to dry, and then we'll come back and show you how to heat fix those slides and stain them. Hi guys. Okay, we're back again. We have some slides on the slide warmer there that are completely nice and dry. You can see there how instead of a glossy kind of drop of water on those slides or drop of liquid culture on those slides, you have a whitish kind of haze and it'll look dry, it'll look like there's no more liquid there, and that's exactly what you want to see on a smear after you finish the warming evaporation of liquid phase. So we are good to go. So we're going to go on to heat fixing these and then move on to staining. So Sabrina's going to show you how to hold these guys for the heat fixation step. So there's a little slide holders there that clamp on each side of the slide. They're a little fiddly to use, but they can get a good grip on each side of the slide, and this lets you pass that slide through the Bunsen burner without getting your fingers in it. There's also these clips that work well as slide holders as well. They're much more intuitive to use. You just clamp them at the end of the slide. Usually you always would clamp where your tape label is, and we still will show you how to kind of label up these slides even though we did it in advance. So we're going to get that Bunsen burner going. There it is. Sometimes they're always a little bit fiddly about starting. And all we're going to do here is pass these through the flame twice. They're going to go through once, you're going to flip it over and come back through. And now that's all it takes to heat fix it. You don't want to or need to hold it in the flame for minutes at a time. It literally passes through once, one direction, and then back the other way. And those bacteria now are killed on that slide, but they're kind of stuck and fixed in place so that their structures are still intact enough that when we look at them under a microscope, we will see bacterial cells. So at this point, we're going to start with one slide. We'll just show you one slide being stained. And we're going to use this nifty tray here. So it sort of looks like a baking pan with a bit of a rack in the bottom of it. And this lets you put liquid onto those slides. As we've discussed in the staining process, you're going to basically bathe those fixed cells with various dyes and solutions. But lets you do that, and then it's just going to settle in the bottom of the pan, and then we can clean that out at the end of the lab. I'm going to zoom in enough so that you can even see that. You can even see that little smear that's there. And you'll see too, when Sabrina's hands come in, that she has gloves on. So the stains stain your hand. They're not ultra toxic, but you do not really want that staining your hand. So it's good to wear gloves when you're doing staining. With other organisms in the lab, we're not as concerned. So what Sabrina has done is she's dripped enough crystal violet dye. You can see the bottle there, and these will be available to you in the lab when you're using them. Enough crystal violet dye to completely cover. You do not want to skimp on dye for these. Completely cover your smear. You don't have to cover your whole slide at all, but you need to cover that smear really well. Then you're going to count to a minute. So the staining times for these processes are not ultra precise. You do not have to time this to exactly a minute, and it won't work if you go 3 seconds over. So I often, and I think that's what Sabrina's doing, I just count it out in my head. And many of the stains, about a minute is going to work just fine. So once we've got to a minute of this crystal violet dye, she is going to wash this off with water. So you hose from the top of the slide, you hold it on a slant, and you wash and wash and wash all the way down the slide until it's free of purple coming off that slide. You may see a faint trace still, especially actually around where your smear is, but you really want to not have puddles of any of the dyes in these steps left on the slide. So at this point, we have a slide that is essentially a simple stain. We have used one dye. It's not doing it. It's going to label all bacterial cells that are in that smear. It's not differentiating them in any way. And if you were to go forward at this point and all you were doing was a crystal violet simple stain, you would place that slide in the bibulous paper sheets to dry it off. And you can see Sabrina's even just going to demo this right now. Because if you were doing a simple stain, which you'll do in the lab, you would move straight on to drying it. Do not confuse that bibulous paper with what is also called lens paper. Similar looking, totally different paper. And you can see now that the excess puddles of water that were on that slide are gone. So you don't want that there when you go forward to stain. So you blot it off with that bibulous paper. And once you're done with that, you're ready to actually go and stain. Today, or go and image that with a microscope. Today we're going to keep going with the stain. So you've learned also about differential stains, and you've learned about the Gram stain, and you know that crystal violet is the first step of the Gram stain, and you also know that iodine, which is what is going onto our slide right now, the mordant is the second step of the Gram stain. So Sabrina's going to keep going now, and we're going to continue this process to give you, show you how a Gram stain is done. It's very simple. Once you know how to do a simple stain, they're all very simple to do. So right now we're going to wait a minute for the iodine to help really bind that crystal violet to the bacterial cells that are in that smear. All of the reagents for this, when you come into the lab to do staining, and I'll just pan over to them, they will be available to you all lined up nicely, the different dyes you need. Back there you can actually see the bibulous paper packs, and the lens paper packs are in a little beaker back there. Your water bottle and other bottles that you need will be there. And everything will be available at your station in the lab. Back to here. Like I said, about a minute is great. You can see the iodine has that characteristic brown color of iodine. Rinse it off just like you did with the crystal violet. Make sure you don't have clumps of iodine left there. Rinse, get it nice and clean. You don't want, if you have too much dye on your slide, you end up looking at dye crystals and chunks of dye when you're actually doing your imaging, and it's very distracting and can interfere with you being able to see the bacterial cells. So rinsing is really important. At this point, you're going to use that, what's called Gram decolorizer. It's essentially an alcohol that, well, I'm not going to give away what it does, but you should know this from the Gram stain. It's a very, very important step of the Gram stain. This is what is actually going to turn this stain into a way to differentiate between Gram-positive and Gram-negative bacteria. So you're thinking in your head, everyone's thinking, right, that's how that's working. I know what's happening right now to those crystal violet stained cells that are in that smear. That's a quick one. So key thing here with the alcohol, the decolorizer, is that it's only 10, 15 seconds. And you might find that you go either a little bit less or a little bit more as you get expert at doing staining in the lab. Because you really need to not leave that on too long. So if you leave it on too long, everything is going to look Gram-negative. And if you think about how the stain works, you should understand why that happens if you would leave your decolorizer on for, say, 2 minutes. Now we're going to add our final stain. This is the safranin stain. It's a bright red stain, or the dye is bright red. It's going to leave a pink color, so the cells themselves are going to look kind of bright pink after you've used this stain. And this can be used as a simple stain. It's just a single stain for cells, but as part of the Gram stain, this is what is going to label up your cells that may have lost their crystal violet dye after the decolorizer, so that you actually visualize those as pink. Those are your Gram-negative cells. I'm giving away some of those steps. So we're going to do that safranin for a minute in order to basically take our colorless Gram-negative cells post the decolorizer step, give them a pink color so we can actually visualize them. And we'll say there's a minute up, and we're gonna rinse it. So same kind of rinsing process at the very end as well. And then you're going to go to that step at the end of the gram stain, like we showed you at the end of when we were sort of doing the crystal violet only stain and showing you what a simple stain would look like. You're gonna blot it with bibulous paper, make sure it's dry, and then you would basically at this point, get out your microscope or pull your microscope over, plug it in, get it turned on, and start your process of actually visualizing these cells and getting them in focus on the microscope. And then maybe just at the end here, we'll go over, because it's actually easy to see there, we'll go over how you label your slide. It's like anything in the microbiology lab, you have to label things. And in this case, you want to be labeling with the obvious suspects of the date, your initials, and what you are staining. So there you have July 10th, 2020, SC for Sabrina Cornish, and E. coli broth for our source. And that goes on as a tape label at the end, because if you label directly on the glass slide and you get even a splash of alcohol on that, it's going to take your pen off. You want to be careful still, though, that you don't spray alcohol decolorizer or the other dyes onto that tape label, or it's going to either wash the pen off or obscure it. So that's why you usually use that. You hold that and then you rinse the rest of the slide down from that tape label. Anything I haven't colored? Oh yes, Sabrina is going to show you. So I'm going to zoom back out. She's got the pan there. The very last step obviously is clean up, and you're going to go to the middle sink. It's a little hard to see, but there's sinks, as you know from working in the lab at this point. Those sinks in the middle that you haven't used until this point are what you dump your dye into. So the end sinks are white, and all those beautiful colored dyes are going to turn those sinks purple and pink, and they're actually quite hard to scrub off. So we use these black sinks in the middle, and at the end of each staining session, you will rinse out that tray just with tap water. There's taps running down into those middle sinks. Rinse out those trays with tap water, tap on any excess liquid, and let them dry on your bench. And at that point, like I said, you would go on to doing your imaging. You're all done with staining. You notice too, one last thing just to mention, that we didn't use a Bunsen burner. After we did the heat fixation step, we don't need to use a Bunsen burner anymore, because we are not concerned about working with live cultures, and therefore we can do all of our staining without a Bunsen burner on. Anyway, thanks Sabrina, and thank you guys for watching this, and that is the end of our gram staining video. Thanks.